Good morning. I'm glad to see so many people here at the first talk of the morning on the final day. I'm going to talk about social evolutionary theory and applying it to cancer progression and also understanding cancer susceptibility. We've already sort of talked about why we apply evolutionary theory to cancer. One of the reasons that I think it's particularly valuable is that it's a system that's really governed by the same kinds of underlying dynamics that we have in lots of other biological systems. And in particular, I think the fact that cancer cells have to deal with resource dilemmas just as any organisms living in an environment with limited resources do is particularly important and informative for understanding why social evolution applies to cancer. So we're sort of going to talk about two kinds of questions today. One is, how do you build an organism with multiple cells? It's not really trivial, as you may have sort of gleaned already from understanding a little bit about how cancer works. And also, if you know anything about social evolution, you know that it's pretty hard to get cooperation to evolve when there are selfish interests. And then the second question is, how do social and competitive cells evolve within the body? So as, even though we've gotten this large-scale coordination in order to create a multicellular body, we're still dealing with the emergence of both cooperative and competitive phenotypes in cancer cells that can be problematic for the entire organism. But first, we're going to start with a little recap of the game that we played 2 days ago. So I'm going to just go over again real quick how we played the game. So if you remember, there were two ways to win. One was being a healthy, uniform tissue with no spaces in between and everybody being continuous. And the other way of winning was a team win, where one of the teams managed to metastasize to the back row and then get to the door, which essentially getting into the bloodstream. You were able to do three things, right? You were able to divide if you had adjacent seats next to you or if both of your neighbors on either side gave you permission. You were able to move one seat in any direction if you had two neighbors who both gave you permission. And you're always able to kill neighbor anytime for any reason. So unfortunately, I forgot to take a T0 picture. So this is after 1 minute. And I just want to point out one thing. I don't know if you could see Andrew Reed pointing towards the back there on the red team. Some strategizing during the break, I think. So this was after 1 minute. This was after 2 minutes. And I don't know if you recall, but I told folks that if there was more than one cell in a seat, you had to be standing. So we have, the red team already in the first time period, there were some standing folks. And here they're getting pretty close to the back of the room. Not much has changed in the rest of the room. By this point, they're fairly close to the back row, and then this is what happened at the end. And in case you want to see what was going on, since I know we're all sort of in our own little worlds as we were trying to divide. This was the last 45 seconds of the game.
Once you reach the final row, you can go out the door. All right.
I don't know if you could hear that, but Steve Stern says, we're in the bloodstream. Blood, 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 blood at the end there. So.
Left row. So we have a winning condition. Red team has won.
All right, so this is our plot of what happened to the clone sizes over time and what happened to the invasion. So as you can see, the red team by the first time period was almost half of the population, maybe about a third of the population, and then by the end they were more than half. and they were more invasive, twice as invasive than any other of the colors. So I just want to kind of ask everybody as a group, why do you think the red team won? Yeah, actually, let's see if we can get these passed around.
I think they cooperated aggressively.
They reproduced at a very fast rate.
They killed at a very fast rate.
They also started closer to the bloodstream.
I heard that they cheated.
Steve?
That's the point.
How did the red team cheat, if you are admitting to such actions?
We decided to kill immediately.
This is an observation from another team, and that is that even after the first minute, somebody on my team said to me, "Watch out for that red team, they're deadly." That was like after the first minute. Yeah.
Anybody else, either from the red team or from neighboring teams?
It wasn't just that the red team killed, that they didn't kill each other generally. They went after somebody else. So there was cooperation. There was some killing, but most of it was, oh, I heard, oh, we're red team, go kill somebody else.
Anybody else? Yeah.
Is it that red is a more aggressive color, so they kind of imbue themselves with a more aggressiveness?
Somebody else suggested that, actually. What strategies did you all play, whether you were red or not red?
So I was leading the red team, and I was not sure. I didn't want to wait to see what the other clones were going to do, So I figure maybe they're going to kill me. So that's, I start just multiplying and killing. I mean, the goal was to win, right? So, as a team.
I was very conscious of local cooperation amongst the red players with a very aggressive aim.
On the violet team, we were just trying to escape the red team and not die.
So I was on white, which is number 2, and I think it's fair to say that we didn't know what we were doing, and that still came in number 2. So that raises the question then, what did yellow do to lose?
Any other thoughts about strategies? We talked, oh, go ahead.
Because I don't think the leader of the pink team was here. I was his only clone, which was, the strategy was probably to stay put.
And how would you play differently if we played this again?
I would cooperate with those who cooperated with me.
Or so
Anybody
you
else?
say.
I would have killed and then recruited right away.
All right, and what did you learn, if anything, from the exercise?
Selfish behavior seems to be prevalent or a good strategy.
I was surprised about the cooperation aspect, but also how quickly the resources become limiting. It was a question of getting people back from the sides, and that became very stressful.
I'd learned that as a teaching method, I think is effective. I've heard a number of people say this is going to stick with them, whereas some of the lectures will fly by.
So I indirectly learned don't count on the niceness of strangers, because I had this idea that was in front of me was empty, so I figured I'd go there and recruit people, and instead 2 people moved in and both killed me at the same time. So, so that was my lesson.
Yeah, I was struck by how quickly it became pretty impossible to get the red team under control by the rest of us.
A couple more comments in the back there. Did anybody else have? No? Okay, great. One more then.
I'll just add one. So I happened to be sitting at an edge, and it was striking how constraining that was, you know, how little you could do. And so some geography is probably important, and that's probably a flip side of the same thing about killing people, because that essentially gives you space.
Great. So I want us to spend a little bit more time doing a group activity. We're going to try to limit this to about 3 minutes of discussion, but I want you to get into groups of 4 or 5 people, and the challenge is to think like an immune cell. So if we were playing this game again, and I assigned a couple of you, your team, say, to be immune cells, and your way of winning was by keeping the tissue from metastasizing for as long as possible. What strategy would you use? And if you could change one of the cell rules, what would it be? And these are the actions available. So if you could mutate one of these rules, or not necessarily mutate, but change one of them as an immune cell, what would it be? And how would that help you to keep the tissue under control? So group up, groups of 4 or 5, and discuss, and then you'll just state what your strategy is and what your rule is.
Good question of clarification.
Yes.
When you change the rule, does that rule only apply to the immune cells, or
Only applies to the immune cells. To all share our strategies and which rule we would want to modify. So let's start over here with this group. Yeah, over here.
So I guess our strategy would be called tumor smacking, is what and the idea is if you have a bunch of immune cells and a tumor cell hanging out, that the immune cells just keep smacking the tumor cells. So you don't kill it, so that way new neighbors can't move in, but you just annoy it so much that it's stunned and can't divide. So you just pester it into just hanging out there. So, yeah, harassment. Yeah.
Are we doing this rule tweak also?
Yeah, and would you change a rule for?
And then for the immune cells, we would change the rule, the moving rule, so that they could move around more freely and monitor more cells.
Great. All right, maybe up here next? Oh, yeah. Oh, great.
So that was kind of our move. So do you want to pick a different one?
What's that?
Oh, because they were talking about moving. Okay, I'm sorry. I thought that they used our move or our modification. So we were talking about it would be beneficial if we had the ability to divide more. So we would change the rule so that we could divide in any direction, not just left and right. And that would give us the ability to maybe overwhelm the tumor more.
Okay.
With no permission.
We were a group of 2. We understood that we were allowed to change a rule just for, that would only apply for immune cells.
Right.
The rules would stand for all other cells. So we chose to change the move rule, and we would like to see immune cells be able to move almost like chess pieces and maybe in a diagonal only or something like that. So then we could get ahead of the out of control cells and at least stand a chance to kill them.
Great.
Like part of his insight, I think, is that it's great to have a rule about controlling somebody else's division, but it only works if you can get there. But if you can move more than one space, now you can get a lot of places and control a lot of territory.
You guys want to say what your strategy was going to be?
I'm sorry?
You want to talk about what your strategy was going to be?
Oh, we didn't get to.
Well, it sounds like you're saying to get around.
Yeah,
yeah.
Otherwise you get stuck in some local non-optimal thing and you can't get out of it. But if you can move 2 or 3 spaces, now you can control his division.
Right. Great.
So if that group had the smacking rule, ours was more of a police state type of rule where no one is allowed to move without our permission, and if they do, we kill them. Yeah. Everything will be under control.
And did you change, which of these rules would you change?
Well, it was the can kill neighbor at any time for any or no reason, and we said there need to be a reason before you could kill someone. That's right.
One of the rules that we came up with was in the divide section. So let's say we recognized a certain color and killed that color, then you could rapidly divide and expand based off of that, because it's like you're detecting a certain antigen and then it's expanding based off of that antigen.
Though we were plotting to take over the entire world with a T-cell lymphoma line.
Any other groups?
We'd like to be kind of more of a surveillance group, so kind of be around the perimeter, but then able to move in when we see something's going wrong and kill them.
Great. Okay, so what I want you to do now is, if you were the spokesperson, I want you to, is there one more? Okay. Oh, and you guys haven't spoken yet either? Okay.
We took the draw the line in the sand philosophy, so any cell that got beyond this point we're going to knock out. And the rule we were going to change was that we didn't need permission from adjacent cells to move.
Okay. Back? One more here. Yeah.
Yeah, so we were interested in the immune cells being able to increase the adhesion of the tumor cells. So that was what, and then we were going to change the movement rule that you had to have, I guess it was for the immune cells, had to have more permission than what's there. So movement was what we were focusing in on, both through adhesion and then more rules before you could move.
So you're tweaking the rule for the other cells to make them require more permission, like all
Yeah, I
four
think
neighbors or something?
That's really what we were talking about, right? The other cells?
Yeah, we started off, I think, not realizing that we were supposed to conceive of our immune cells as having evolved from this state of rules. And we just wanted to focus on things that were moving and kill anything that moved.
Okay. So if you were the spokesperson or somebody else in the group, write down what that rule is that you would change for 2 reasons. One, I want to collect them for future versions of the game. And the other is for the next thing that we're going to discuss. So get out a pen and a piece of paper and write that down.
And let me know if you want me to go get a pad of paper for you.
I'm going to give you a couple more seconds to write that down. I want you to have it clearly articulated. Yeah? All right. Everybody all set now? Anybody need a little more time? Okay, so what I want you guys to do now is, given that the immune system is going to follow the rule, which you just specified, I want you to think like a cancer cell that's trying to get around that rule. So what strategy would you use now as a cancer cell, given that the immune system has the ability to do the things that you specified and is using the strategy that you specified? And what one rule would you change again? And how would you do that? So both the strategy that you would use, given that the immune system is using the rule which you just wrote down, and what rule you would change if you could change a rule.
So your strategy can depend on changing the rule.
your strategy as a cancer cell. Yeah. Yeah, so here we're assuming that you're a cancer cell that's in the environment where the immune system is behaving in the way you just specified. Now, which of these rules would you want to change in order to metastasize, given that the immune system is behaving the way you specified? So here, you can only change your rule as a cancer cell. You can't change the immune system rule. Yeah, so now we're assuming the immune system rule and strategy is fixed based on what you just specified. What your cancer strategy is and what the rule is that you'd break.
Okay. So our immune system rule was called tumor smacking, where the immune cells can move around anywhere and not kill the cancer cell, but just harass it by smacking it. And so our cancer cell rule we called, I didn't come up with it, called smackback, which is now if the immune system cell smacks the cancer cell has evolved to actually now like smack back and electrocute the immune system cell and kill it. And so it's sort of like harassment over time. It goes back and kills the harasser.
Okay.
So.
Where's our second mic? Oh, wherever it is. Yeah, great.
What we think the cancer cell should do is basically...
Well, tell us your immune system rule one more time.
Oh, our immune system rule was that the immune cells could move freely and the other cells would need more permission to move. Okay. So the cancer cells, we decided, would basically remove any cell surface receptors that they have for immune signals, basically becoming deaf and blind, and they would create a new cell surface marker, which Carol's going to show you.
And Dr. Stearns has just returned to the room, but this was the new cell surface marker, which he didn't show it, but this is how we translated it. Okay. That would be coming from the cancer cell to the immune cell.
Next team.
Okay, so our immune strategy was to have kind of like a policeman immune surveillance around the perimeter that could move in and target any cells getting out of hand. And our cancer strategy was one of bribery and corruption. So any tumor cell that came near to us, we would kind of bribe or signal to them and corrupt them to be on our side, so not to kill us, but maybe to kill our opposition.
Nice.
Or become one of us.
Our strategy was, as immune cells, was to move large distances and sort of like keep an eye on the door. And the counteraction by the cancer cells was basically just to divide your way out of that so that the immune cells couldn't keep up. So the rule was under divide into adjacent seats, left and right, we would extend that to backward and forward as well.
Great.
Our immune rule was that we could increase the rate of division in response, non-linearly in response to the number of cancer cells there were, and our rule for escape is that we would be able to move anywhere.
So our rule, which we imposed on the cancer cell as the immune cell, was you can't divide without permission of the immune cells, and if you do, we will kill you. So that was the police state rule. And then the strategy from the cancer cell was to basically get rid of this idea of you need permission from both neighbors. So we would divide from the inside and just divide so rapidly that those cancer cells on the outside would get killed because they were getting pushed forward without permission, but everyone on the inside would be okay. And then the other alternative strategy to that was just get permission for false reasons and try to convince the immune system, oh, no, we're really dividing for good reasons.
Okay, so our previous immune strategy was that we would divide in any direction without permission. Well, that was the rule, and the strategy, in a sense, was to stop aberrant proliferation. So if we were cancer, what we would do to adapt to that is we would actually stop killing our neighbor. We would recruit them to become part of our group. They would come to the dark side, essentially.
our group's immune cell rule was that nobody could tell us what to do. We didn't need permission. We didn't need permission to move. And then our cancer cell rule change was that our neighbors can't kill us. So we would mutate and change so they couldn't recognize us. But then they'd still be hampered by the permission to move. So we're sort of a gentle tumor, slow growing.
Great.
We had another strategy, which is that we're going to make the immune system think we're one of them. So we're going to mimic the immune response and can't be recognized. Since, you know, the other thing was that the cells couldn't get past a certain point.
Great. Other groups? Oh, excellent. So let me now take us through a few facts that I think will help connect a little bit with some of the experiences that you guys have. So as we know, most of us know at least the basics of social evolution, right? That there are a lot of traits that individuals can have, including here cells, that can have effects on the fitness of others. And as you saw in this game, if you're following some simple rules that mimic aspects of cancer cell behavior, there's a lot of dependence on your neighbors for your fitness. And whenever you have a situation where traits affect the fitness of others, you can get selection acting on those traits with that have social effects. And that's really the process of social evolution. And through that, you can get selection for pretty complex phenotypes, including things like production of public goods and other types of cooperation, division of labor, complex communication systems, and also the emergence of higher level entities. We also know that population structure can influence selection. I'm not going to talk about that in too much depth because that would be a whole hour, but that relates to a number of other systems in evolutionary medicine as well. So these are sort of the two main questions. Remember how multicellularity initially evolves and how it breaks down, and then also how cancer cells evolve their competitive and social phenotypes. And cancer cells have a variety of behaviors that map on to some of the things we were sort of playing with over the last couple days in here. So one of them is contact inhibition. This is the idea that cells, if they have neighbors, will not divide. They're inhibited by being bound to their neighbors. Cancer cells also pretty much always, cells, if they're normal, require external signals for proliferation and survival. And those systems often get altered in early stages of neoplastic progression. That cells start being able to signal to themselves, give themselves permission to proliferate, give themselves permission to survive, give themselves permission to move, all of those things. Similarly, there's a phenomenon called anoikis, which is basically that cells, the program is that if you're not attached to neighbors, you die. And cancer cells will alter that so that they can just kind of go off on their own. So that would be analogous to one of the cancer cell groups saying, well, the rule that I'm going to change is that I don't ever need permission from neighbors to do anything, basically, and that you could go off on your own. I kind of imposed it by saying that all of these rules that you had to follow were dependent on having neighbors. It was sort of indirectly imposed on you that there was no way to do anything other than to be attached to others. The production of growth signals. This is not just the production of signals for yourself to grow, but also the production of signals that may promote your clone or the other cells that are nearby, whether they're the same clone, a different clone, or maybe they're host cells like fibroblasts that you're trying to promote because they're promoting you. So a couple of you guys sort of hinted at strategies where you're, as a cancer cell, trying to co-opt the immune system. And we know that one of the things that cancer cells can do pretty effectively is co-opt systems of the body, whether it's the immune system or other cells that are nearby. That seems to be a pretty important factor. The production of angiogenic signals, that's another interesting case. I'll talk a little bit more about that later, but that's basically the production of signals that make blood vessels grow. So it's essentially recruiting more resources that can then be used to divide more rapidly. And I already mentioned the immune system predation and eventual co-option. There's an interesting process that if we did a few more rounds, perhaps, of thinking like a cancer cell and then thinking like an immune system cell, we might have ended up with cancer cells that were able to get the immune system to promote their own proliferation and things like that. Very importantly, there's a whole lot of research now on the microenvironment. So if you look in the cancer literature and you search for sort of microenvironment, you'll see lots and lots of papers. And what this really means is that the fitness of the cancer cells is being determined by the other cells that are around, and not just the cancer cells, but the stromal cells and other cells that are nearby. So it's known that cancer cells, if you take cancer cells and put them in a normal microenvironment, then they can act like normal cells. They can produce ordered tissues, and that's really been surprising, that you can just take a cell that's really messed up, put it in a normal environment, and all of a sudden it's acting normal. We also know that you can sort of do the opposite. You can add sort of environmental cells here, activated fibroblasts, and they can induce normal cells to act like cancer cells. So there is a very important interaction with the social environment, in other words, the other cells, that really determines whether a particular genotype will end up as a cancer phenotype. So when did cancer start? Carlo talked about this a little bit. It began at the dawn of multicellularity. And multicellularity is really sort of the ultimate in social evolution, right? We have all sorts of traits that are essentially affecting the fitness of the other cells that you're teaming up with to make this multicellular body. And there's a sort of general principle that occurs when you're pooling your reproductive interests. So the somatic cells basically curb their reproduction. That allowed for just the germ cells to propagate and the somatic cells to really kind of focus on how to make the entity more viable, dividing labor and pooling the resources, and really creating a behavioral phenotype that was for the benefit of the organism. And cancer is sort of breaking that covenant. The cells start promoting their own fitness, as some of you did in the game, especially the red team, and monopolizing the resources, and potentially also not doing the activities that are required sort of the cells as a part of a multicellular entity. And this can have a really detrimental effect on the fitness of the organism. So on the surface, it seems really obvious then that in order to evolve a multicellular body, you need to suppress cancer, not just because you need to curb proliferation, but if you're going to have a sort of aggregate level phenotype, all of these cells behaving in a way that will promote the fitness of the organism, you need to have pretty tight controls on the kinds of things that you guys were doing as cancer cells, like moving all around and dividing out of control and all of that. And failures in cancer suppression really sort of lead to the breakdown of the multicellular systems that are in place to allow our cells to function as one concrete entity that can do stuff. And we can kind of extend this then to say, well, there's the question of how do you make a multicellular body, and then there's a question of how do you make a larger and longer lasting multicellular body. And it's really sort of a continuation of the same problem. If we figure that there's some risk for getting cancer that's on a cell-by-cell basis, so some risk per cell per year, then it should be that larger and longer lived organisms get more cancer than smaller and shorter lived ones. But that's not the case. And in fact, if we look at across species, it's often the species that are larger that seem to have better cancer suppression and lower chances of cancer. So this is Peto's paradox, which is why doesn't a very large animal that's, say, 100 times as large as another animal get 1,000 times more cancer? And this is some data that Aaliyah Collin and Ken Yan have pulled together, showing that it really is not the case that cancer incidence increases with body size and lifespan. In fact, it looks like there's a slight negative correlation there. And this is because evolution is really kind of optimizing the trade-offs here. So for a mouse, there's no advantage to suppressing cancer any longer, then they're likely to live because of external mortality. But for an elephant or a human, suppressing cancer really can increase survival to reproduction and also the capacity for parental investment. And so then we can ask the question of how does evolution suppress cancer in these larger and longer lived organisms? And again, it's clear that cancer mortality in humans is probably higher than in much larger animals like elephants. There may be a lot of other reasons for this, but it's certainly not the case that being larger makes you have more cancer. So Carlo has been working on this question and has found that elephants actually have more tumor suppressor genes. They have apparently 12 copies of P53. And he could sort of tell you more about this, and they're trying to work out all of the details of how this is the case. But it's a really interesting finding, and they're sequencing the humpback whale now to look at whether the humpback whale has more tumor suppressor genes as well. I'm going to talk for just a minute about the question of whether unicellular organisms have cancer and what cancer would be like in a unicellular organism. And just sort of on the surface, it seems as though you can't really have cancer if you're a unicellular organism because it's a phenotype that requires cells to be not following the rules of the larger organism. But it is the case that there are unicellular entities like yeast and bacteria that form biofilms and dictyostelium, slime molds that have aggregate phenotypes, and those phenotypes can get disrupted by individuals not following the rules, like proliferating too much, et cetera. And do these constitute cancer? Probably not, but there's certainly some parallels there. So we're talking about more general principles that can apply not just to multicellular entities, but potentially to other entities that may be unicellular, but have some aggregate phenotype that can get disrupted. So bottom line is that the process of social evolution can shape cooperative and competitive phenotypes. And Steve has a question there. Do we have a mic nearby?
Eukaryotic cells actually have a hierarchical structure like a multicellular organism because of the organelles, which have come in with their own independent genomes. And yeast get mitochondrial cancer. The petite mutation in yeast causes a mitochondrial cancer. It has all of the effects on the aggregate like a cancer has on a multicellular organism.
Very cool. Thanks for that.
Yeah, I've asked, I've asked pathologists if you see mitochondrial cancer in human cells. Do you see cases where the mitochondria are dividing out of control and killing the cell? So at that same level, and they basically don't know. There's some choriocarcinomas that seem to be packed with mitochondria, but they couldn't tell me whether that's killing the cells. But the yeast example is a great one.
All right, I'm going to jump ahead because I've only got about 10 more minutes. And I just want to point out that there are important sort of social dilemmas that cancer cells have. So normal cells are really programmed to use resources and reproduce in what you could call a sustainable way. So they're not trying to get more and divide faster. But cancer cells can use resources faster, create more waste products, replicate more quickly, recruit blood vessels to provide resources, suppress the immune system, invade the bloodstream, colonize new areas where they can get more resources. And it may very well be the case that premalignant tumors are self-limiting due to resource dilemmas. So they may divide a little bit, but then not have enough resources to continue. I think these are open questions about why some premalignant growths are not problematic. And sometimes they grow and then they disappear. We don't necessarily understand that process well. And another important point is that these resource dilemmas change the selective environment for cancer cells. So there are a number of limiting resources. These are some of them. There's probably a lot more. It's not very well understood yet exactly what all of the limiting resources are. They may be different for different kinds of cancer cells at different points in progression. And one of the things that I've done is applied dispersal theory to understand the evolution of metastasis. So does dispersal theory, can we apply that to understand why cells evolve to be motile? And we know that cancer cells have a lot of ways that they can move around. And we started out just by modeling probabilistic movement. Basically what we found was that intensive resource use on the part of cancer cells, so having a high rate of using local resources, a high metabolism, essentially, that led to higher rates of motility among those neoplastic cells that were using resources at a faster rate. But the normal cells retained sort of normal rate of motility. So to us, this suggests that resource dilemmas may drive some of the process of invasion and metastasis. Likewise, these limiting resources are not necessarily fixed. So if you have individuals consuming resources, then they're changing the resources that are available for others. And we know that hypoxia, so the lack of oxygen, that's patchy, and it changes over time. And local cells, if they're growing too quickly, they can occlude blood vessels, and new blood vessels can grow. So this can be a very dynamic process. And Carlo and some of his colleagues did a model of the evolution of cell migration when you do have vessels being occluded and new vessels growing. And basically what they found is that the more spatial and temporal heterogeneity in resources, the more migration there was. And this was similar for both random migration and for gradient ascent motility. So what if we look at the clinical literature here? Well, it turns out that if you take wild type mice and give them xenograft tumors, they can produce these sort of abnormal vessels, which lead to heterogeneity in the availability of oxygen in these hypoxic regions. But there's a genetically engineered mouse that can have normal levels, normal vessels, even when they're given a xenograph, and that leads to more homogeneous resources and less hypoxia. And it turns out that in the mice that have the more normal distribution of resources, because their vessels are normalized, that there's improved perfusion and oxygenation of the tumor and significantly lower rates of metastases in the lung and in the lymph nodes. So these questions that are really very general about ecological theory and dispersal and how resource heterogeneity can affect things like mobility have really important applications to clinical issues in cancer. And I think this is just the beginning of a whole set of possibilities for applying ecological theory to cancer. And it should be noted that the folks who published this paper, they weren't thinking in terms of evolution and dispersal theory. They just found this. But of course, it fits very well within the ecological framework of dispersal theory. We also know that hypoxia in primary tumors is associated with metastasis. And further, that the global dispersal, this metastasis process, occurs rapidly after the local dispersal, so just the invasion into neighboring tissues. So there's a kind of direct line from resource competition to the evolution of cell mobility and metastasis. And like a lot of organisms do, I think cancer cells can really create a niche in the body to promote their own survival and reproduction. So they are able to basically take what I think we often think of as a passive resource delivery system of blood vessels and manipulate them. And they can affect them in two ways. So one is that the blood vessels can be destroyed if the cells are growing too much, so that's one social effect. But they can also grow if the cells are signaling for them. And this is really an analogous process to the creation of irrigation systems. Very, very similar. And one of the things that is very characteristic of tumors is that they have very inefficient resource delivery networks. So instead of sort of nice, neat, fractal resource delivery networks, they have very tangled and inefficient and leaky ones. And we think that it's possible that one of the things that's going on here is that the tumor cells, rather than signaling cooperatively with the other cells in ways that can most efficiently distribute the resources among the cells that are there, they're instead signaling to try to get the most for themselves. So in other words, the upstream cells may be kind of opening up the taps for themselves with angiogenic signals, and that may starve the downstream cells, which could be part of why we see hypoxia in the middle of tumors. But this is, of course, an area that we need to do more research in to find out exactly how these influences occur, at what scale, and whether this theoretical framework is the appropriate one for understanding how angiogenic signaling occurs. So in my last couple of minutes, I want to talk about what I see as one of the frontiers in applying social evolution to cancer, which is how does human social behavior affect cancer susceptibility? So now we're kind of going to a totally different level, which is how does our capacity for social interaction and the kind of things that we're affected by, how does that influence whether or not we're likely to get cancer? And the reason that I see this as a frontier is because we're just starting to scratch the surface in terms of the co-regulation of behavior with hormones. So we've known for a long time that hormones affect behavior. We're just starting to realize that behavior also affects hormones. And of course, it's been known for a long time that hormones affect cancer susceptibility. So I think that there's a really large potential to be applying our understanding of social evolution, or of social behavior, and co-regulation of hormones to understanding cancer susceptibility. And I'll give you just a little taste of some of the work that I'm doing. So one thing that I think is particularly telling is that there's been a pretty recent study showing that psychosocial stress actually leads to faster ovarian depletion. So the oocytes actually get used more quickly. And antral follicle count here is the number of oocytes that are sort of competing in order to be the oocyte that goes off. So these things are, to me, indicative of a faster life history strategy, but they also indicate that you're going to have a different kind of hormonal profile because it's the oocytes that are producing most of the estrogen and then the after the oocyte leaves, what's left produces a lot of progesterone. So there's, I think, a very clear link here between social input and social behavior and hormones that could have a very important impact on cancer susceptibility. And I want to kind of make a call back to some of the things that Steve was talking about earlier in terms of the the basics of life history theory, that uncertain environments can lead to selection for reproduction at the cost of somatic maintenance. And it's also the case that early adversity and stress may be providing cues that shift organisms towards reproduction at the cost of somatic maintenance. And we've been looking at breast cancer sort of from this perspective, and it's starting to look like the hormone receptor positive breast cancers may be a result of environmental mismatch, where you're getting a lot more hormone exposure, cyclical hormone exposure, because of having more cycles. And we're seeing higher risk from lower fertility. But the hormone-negative breast cancers seem to be very different. And we think that there may be life history trade-offs going on here. There's higher risk from, associated with higher fertility. And one of the other things that we're looking at is whether early adversity and stress may be a risk factor in hormone receptor negative breast cancer. And I'm just going to kind of show you a couple of slides here that we found that breast cancer is associated with certain early life stresses, like being in the Holocaust, being in the Dutch famine, the siege of Leningrad, other sources of trauma and stress, including maternal death, deficits in childhood, so let me just say that none of those distinguish between hormone negative and hormone positive, unfortunately. So we don't know whether that's specific to hormone negative breast cancer. But we do also know that early life stress affects things like tumor volume in mice, and also in rats it affects reproductive aging, which similar to the recent finding about antral follicle counts and ovarian depletion. So finally, I just want to let you guys know that we're going to have our second International Biannual Evolution in Cancer Conference at UCSF next summer, probably in June. We're just finalizing the dates now, and that will be at the Mission Bay campus in San Francisco. And I hope that you guys all consider coming. We're going to, our general theme is going to be sort of from unicellularity to multicellularity and back again. So what can we learn from unicellular systems, and how is it helpful to think about cancer as, you know, a reversion perhaps to some sort of a unicellular state, and how can we really start bringing in more folks who understand the process of social evolution, who understand ecological dynamics among other types of entities other than cancer cells, and how might those apply to cancer, and giving us sort of more theoretical tools with which to better understand cancer progression and susceptibility, and also prevention. Now, thank you to everybody who's contributed to this body of work, and I don't think I have time for any questions, but thank you.